Hello, everyone. Welcome to the second webinar of this series. My name is Declan Bennett, and I'm the training coordinator for the Science Foundation Ireland Centre for Research Training in Genomics Data Science. So just to kind of give you some background on what the CRT in Genomics Data Science is and what its involvement is with this webinar series. So it's a PhD training program, which is cohort-based. It involves the 7 universities across the island of Ireland, and the aim is to train 100 PhD students over 4 intake periods. So in the first 3 months of a student's PhD, we run a residential training program. And this is where all the students attend the University of Galway, and they undertake training in the fundamentals of genomics data science and its applications to human health and basic research, before then going on to enroll in their final university where they'll complete their PhD. This webinar serves as a kind of a keynote for a week-long set of masterclass training sessions where we have trainers associated with the African Society of Human Genetics on site here in Galway, delivering a wide range of kind of training in genomics data science. So today we have a fantastic talk by Professor Choudhury. Professor Choudhury obtained his master's in zoology from the University of Kalyani in India and a PhD in bioinformatics at the University of Calcutta. He moved to South Africa in 2010 to join the Sydney Brenner Institute for Molecular Biosciences at the Wits University as a postdoc fellow. He's presently employed by SBIMB as a senior scientist and co-leading the AWI-Gen Genome-Wide Association study, as well as the AWI-Gen population structure study analyst teams. He has contributed to many major genomic studies conducted in Africa, including the African Genome Variation Project, the H3 Africa Genotyping Array Design, the H3 Africa Whole Genome Study, and the Southern African Human Genome Project. His key interests are population genomics, genomics of complex traits, and developing computational resources for genomic analysis. So with that, we can begin the webinar.
Okay, so hello Declan and hello everyone. So we are having a very bad internet day today. So, well, can you hear me?
Yes.
So we are having a very bad internet day today. So I have tried to record and share the presentation. So I would request Wilson if he is able to play it, and then I will join in towards the end.
It's fantastic. Thank you.
Afternoon, everyone. Let me share my screen. Okay. Can you see my screen?
Yes.
Okay.
Good afternoon and welcome to today's session. I have planned today's presentation in 2 broad parts. In the first part, I plan to introduce you to the history of African populations and show examples of how this deep and complex history is reflected in the genomes of the people living in the continent. In the second part, I would talk about some of the H3 Africa Consortium studies and how they have contributed to a better understanding of African genetic diversity and demographic history. So let us start with a brief primer on African history. As we all know, humans originated in Africa. However, there is a lot of debate about when in Africa did this happen. Had someone been giving this talk a few years ago, based on findings of some of the oldest human fossils, such as Homo kibish and Herto, they would have probably answered East Africa and specifically Ethiopia. And if the question was when, they would have probably given you an estimated time of around 200,000 years ago. But the finding of a 300,000 years old fossil, Jebel Irhoud from Morocco, has not only questioned this geography, but also pushed the origin estimates further back in time. Also, there are several fossils showing the presence of early humans in southern Africa, suggesting that the human lineage had spread across the continent by about 100,000 years ago. Although the details of the very early human history are still sketchy, over the last few years, some major milestones of early human divergence has been outlined. So this population divergence tree from Karina Slebush and Matthias Jakobsen's review provides a quick summary of our current understanding of early human history. Here, the y-axis shows time, and the width of each branch corresponds to the estimated population size of the corresponding lineage. As you can see, the first split in the human lineage is the separation of ancestors of current-day Khoisan hunter-gatherers from the rest of human population. This split probably took place more than 100,000 years ago. Khoisan is some sort of a blanket term that is used to define diverse groups of hunter-gatherers from southern Africa. These groups are characterized by distinctive appearance, lifestyle, culture, including rock art, and use of click in their language. Although currently diminished and marginalized, it is estimated that in the past, they were spread across most of the southern Africa and probably beyond. The next group to split was the ancestors of rainforest foragers, once referred to as the pygmy, also characterized by unique appearance and lifestyle. This was followed by the split of ancestors of currently East and West African populations. From this East African branch, all currently known Africans are estimated to have originated. And as you can see, there was a strong bottleneck that corresponds to the out of Africa migration originating from the East African branch. So fossils and archaeological records have generated rather detailed timelines and route maps of how the groups of people that left Africa around 60,000 years ago traveled and reached different parts of. A lot is known about what was going on in Africa, which is in like Africa tree between 3,000 and 10,000 years ago. Based on background of ostrich eggshell bits, a study last year suggested the possibility of existence of regional and then trunk 50,000 years ago. However, these networks did not persist for Africa, started breaking down probably around 30,000 years ago, and this was driven by climatic changes. A popular theory is that during this time, the intertropical convergence zones shifted southward, and this kind of led to the flooding of the Zambezi River, which disrupted the route that was used for contact between East and Southern Africa. Similar disruption on other regions is estimated to have led to regionalization of populations evident in geographic region-specific patterns of tools and artifacts. So if you were visiting Africa about 10,000 years ago, you would have surely noticed 4 very distinct groups of people living in the continent. One in South, one in East, one in West, and one in Central African rainforest. And also non-African populations on the regions bordering Africa and West Asia. The innovation of agriculture both in West Asia and later in Africa motivated major surge of migrations within and also from outside the continent. These include movements across the Sahel region and back to Africa migrations. However, the most conspicuous of this was the Bantu migration, the gradual expansion of agro-pastoralist people from Nigeria, Cameroon to almost all parts of sub-Saharan Africa in the last 4,000 to 5,000 years. These migrations changed the distribution of populations quite dramatically and led to this current pattern of peopling in which the Niger-Congo speakers related to the West African branch shown in brown became the predominant group in almost all parts of sub-Saharan Africa. Similarly, the Niger-Saharan and the Afro-Asiatic speakers who are related to the East African branch expanded to various parts of East and North Africa. The Khoisan and the rainforest foragers, on the other hand, diminished in size and became restricted only to certain regions of Southern and Central Africa. So a problem with broad brush maps such as this is that they very often do not provide enough information about fine-scale variation within each geographic region. The ethno-linguistic divisions indicated by a single color in this map includes hundreds or even tens of hundreds of ethno-linguistic groups, each of which probably has a slightly different demographic history. Also, there is another layer of complexity. As you can see on this map of ethno-linguistic distribution of populations from Kenya, you'll note that they are present of several groups, and these groups belong to all three major ethno-linguistic divisions. And you can also note that instead of a clear geographic line, in cases these groups are distributed in an interspersed manner, hinting at a complex set of historical events to have shaped the current pattern of distribution of population, even at a very finer scale. I hope you now appreciate how complex and multi-layered African history is. So far, there is very limited information on how this unique distribution of population had originated. For example, there were probably tens or hundreds of migration events at regional scales, which we do not know about. And unless these are identified and properly characterized, the reconstruction of African history would remain incomplete. Moreover, even the migrations that we know about, there are a lot of debates and questions about their exact routes, timelines, and other nuances. For instance, this map shows two competing models for the Bantu expansion that we discussed a little while ago and will revisit later. So in this scenario, genomics is emerging as a valuable approach to not only test existing hypotheses on recent and deep history, but also propose novel theories and models. In the next few slides, we will go through some of the key features of African genomes and recapitulate some of the major insights generated by analysis of genomic data from African populations. Perhaps the most striking and distinguishing feature of African genomes is the remarkably high diversity. This plot shows the number of SNPs detected in about 1,000 genomes, each from about 25 global populations. Each genome here is represented by a dot, and each population is labeled by a particular color. And the y-axis shows the number of SNPs identified in each genome. African genomes, as shown in the box, on an average harbors about 20% of more variants compared to non-Africans. And this is largely because all the non-African populations went through a strong bottleneck during the out of Africa migration, which considerably reduced their intrinsic genetic diversity. The higher diversity is not only restricted to the number of variants. As evolution had more time to reorganize these genomes, they have much smaller constituent blocks. Although we don't have time for details, but as an analogy, you can think that the breeds that make up African genomes are much smaller compared to those that make non-Africans. So it has long been known that sharing of genes within the family depends on how recently and how many ancestors we share. For example, we share 50% of our genome with our siblings, but only a quarter with our cousins. The comparison of genomes show this pattern of variant sharing, although here we are probably talking about 0.1% or even lower values, extends beyond families to individuals within a population and even the historical relationship between populations. This opens the possibility that with genome scale data, we can model how individuals in a population are related to each other, and also how different populations are interrelated. As each ancestor takes us back further in time, therefore, genome is a kind of a time capsule, condensing the evolutionary history of all its ancestors. And a careful analysis of genomes can not only provide answers to questions about them, but also about their ancestors, such as whether they migrated, whether they met and exchanged genes with other populations, what their population sizes were through time, and also some ideas on the pathogens they encountered and even their diet. So there are several statistical and computational techniques to interrogate genomic data for answering such questions. Of these, the principal component analysis, or PC, is perhaps the most often used method in population genomics. So PC is a general technique that spans almost all the sciences for dealing with high dimension data, which is difficult to visualize and interpret. PCA, when applied to genomic data, can provide an easily comprehensible plot representing relative distance between individuals. So when a PCA is performed, the algorithm is not informed about any labels. The individuals are colored or labeled by ethnicity or other factors only after the analysis. As you can see, so this is kind of a representative or a toy PCA based on genetic data. Here, each dot represents an individual. African ancestry individuals are shown in green dots, Europeans in red, and East Asians are shown in blue. So you can see that individuals from each population tend to form clusters, and the x-axis, which shows the principal component 1 or PC1, depicts the maximum variability in the data. The y-axis, which represents PC2, shows the next higher variability. Normally, we investigate several PCs to get a full sense of a genomic data set. How we might interpret this plot is that it shows African populations, due to their distinctive history, are much different from non-Africans and are separated by the first PC, while the second PC separates Europeans from East Asians. What is more interesting is that if you compare the relative location of these populations in the PC to that in the geographic map, you can see a pattern of similarity suggesting that genes might broadly mirror geography. So let us see how real PCA plot reflects African genetic diversity and correlates with the major migration events. As you can see, the PC1 here separates East African populations from other African groups. The decline formed by the East African population is estimated to be due to the back to Africa migration that happened over the last few millennia. Other African populations appear as a line from west to central west and central to southern Africa, and is probably connected by the Bantu expansion. The separation of individuals in the south from the rest of the cline is perhaps due to the unique presence of Khoisan ancestry in this group. And this is a very basic PC on a limited set of populations, and we will see in a while how the inclusion of additional populations enhances the resolution and also generate more insights. So when a population moves to a new area, it often exchanges genes with the populations residing there, making gene flow events a key marker of migration. We employ a set of specialized tools for identifying gene flow from and into populations. One of the most widely used tools employed for this purpose is named AfSHG. AfSHG firstly tries to model the dataset into user-provided number of ancestries, and then try to detect how much of each ancestry is present in each individual. In this example, data from 5 different populations were provided, and the tool was asked to model the data based on 3, the top plot, and 4, which is the bottom plot. So each ancestral component in the plot is represented by a separate color, and a thin black line separates individuals from different populations. So you can see the European population CUE is colored full in blue, the African YRI in red, and the East Asian in green. Individuals from the ASW, that is the African American populations, is shown here. And you can see, like in the zoomed in version, that it contains both blue and red components, suggesting evidence of admixture of both European and African ancestries. there are also a set of tools such as Mosaic, Molder, and Globetrotter that can then be used to date possible admixture events, providing further insights not only into which groups migrated, where, but also when did it happen. So here is an example of admixture plot based on worldwide genetic data. I won't go into details, but as you can see, there are several unique ancestries such as the rainforest, forager, Khoisan, and Niger Congo that really predominant African populations. And we can also see a lot of Eurasian gene flow in North and East African populations. And as you can see in these maps, the distribution of these ancestries is geographically stratified. And if you remember, it conquers broadly with the PCA and also the migration maps. So finally, let me introduce the third major set of tools in population genetic analysis toolkit. These tools are based on fluorescent theory and models genetic diversity in a sample to the demographic history of the corresponding population. The idea here is to consider all pathways of inheritance through which sampled copies of homologous DNA elements are traced back to a single ancestral copy, known as the most recent common ancestors. These applied to genome scale data can provide insights into estimates of past population sizes, and such temporal fluctuations in population sizes and understanding their causes informs us about how populations have reacted to past events, such as glacial cycles, climatic change, or even large-scale migrations. So using this approach based on whole genome sequence data from just few individuals from a population, we can trace the population sizes of the parent populations in the last 1 million years. So the way it is done is that you break down the genome into segments and then try to find the MRCA of each of these segments and estimate what their population sizes could have been. So depending on frequency of segment sizes of a particular length, you could say whether the population was 5,000 or 50,000 at a particular time point in history. So this is a typical population size dynamics plot. The y-axis in this plot shows effective population sizes. The x-axis represents time in 1,000 years ago. And you can see that these are generally plotted on the log scale. The more right you go, the further you go back in time. So you can see that although all the continental populations have had similar sizes for most of their initial history, indicating that probably they were not separated, around 70,000 years ago, we can observe a severe bottleneck in non-Africans, but not in the African populations. This is because ancestors of all non-Africans were reduced, whereas Africans by and large maintained their population size during this time. Although this is followed by a gradual recovery in population size of non-Africans, what's fascinating 20,000 years is still deeply ingrained in these genomes. The populations, these African groups also show considerable variation with the ancestors of correlation sizes for most of them. In history, actual people who lived at a particular time point. Ancient genomes is making it possible to study individuals from more and more distant past, providing solutions to some of the long-standing puzzles, as well as development of robust models for inferring past demographic histories. So this is a vast topic and perhaps require a talk on its own, but let me just present one recent example. A recent study by Lipson and colleagues published earlier this year had shown that the model of African history that emerges from ostrich eggshell pit is very possible, and indeed populations in different parts of Africa exchanged genes for a quite long time, and which came to an end about 20,000 years ago, which really kind of matches the ostrich eggshell pit distribution. And this kind of, stop of exchange led to the development of regional populations, each with their unique genetic characteristics. So despite the potential for uncovering migration and gene flow and delineating events and factors that shape diversity, there is very limited data on African populations. Just to give you an estimate, the estimate of this level of scarcity, less than 10% of the 2,000 groups have proper genomic data. Moreover, a large number of these groups that have genomic data are represented by very few samples, providing limited scope for in-depth analysis. So there is a need for massive data generation, integration, and collaborative analysis to bridge at least some of the major gaps in understanding of African and thereby human history. So I will now move to the second part. In the second part, I will introduce you to the H3 consortium's studies and data sets and analysis that we have been doing. And so, as most of you know, Human Heredity and Health in Africa, or H3 Africa Consortium, is an NIH and Wellcome trust-funded initiative aimed at generating data sets, developing capacity, and building infrastructure for genomics research in Africa. Over the last 10 years, H3A has set up networks of over 500 African researchers that are spread across 30 African countries. Efforts originating from the H3 Africa Consortium has already resulted in over 600 publications, and several hundred papers are in pipeline. So I have two major connections with the consortium. Firstly, I lead several components of the Origin, a cohort-based study aimed at identifying genetic underpinning of cardiovascular diseases that is nested under this consortium. Also with Prof. Neil Hanchard, I co-chair the Genome Analysis Working Group, which is the forum for planning and implementing cross-consortium genomic studies, and also linking the consortium to global initiatives and collaborations. So today I will be wearing, in a sense, both these hats. As we noted in this figure from our review of H3 Africa publications, although the consortium's work has been largely focused on detecting disease associations, the analysis of the data sets have resulted in several publications that has provided valuable insights into African population structure, admixture, and demographic history. The most prominent of these is perhaps the H3 Africa whole genome sequence study that features as the cover story of Nature exactly 2 years ago. So to bridge the gaps in publicly available whole genome data, our study somewhat strategically sequenced 323 samples at 30x, and also included 220 samples that were sequenced at middle depth or medium depth from another project in the consortium. After quality control, we ended up with a data set that consisted of over 40 million variants and 4 individuals. So the PC of our data integrated with data from external sequencing studies looks like this. As has been observed for African populations in the previous section, the first principal component separated the East African from other African populations, and the second principal component placed remaining population along a line from west to south with a very strong correlation with geographic region. So as you can see, individuals from Cameroon, DRC, and Zambia labeled as CAM, DRC, and PSZ, localizing proximity, forming a somewhat independent Central West African group, with the Zambia sort of localizing at the crux of that line from west to central and then to south and East Africa. This unique localization of these populations in the PC made us wonder whether this has any relationship with the Bantu expansion that we talked about in the first section. The most current genetic database model that was proposed by Patan et al. Estimated a southerly movement of Bantu expansion through Cameroon to Angola before dispersing to south and east. The inclusion of populations from DRC and Zambia provided us the opportunity to explore the potential role of these countries within this important migration route. So when we looked at sharing of a particular kind of genomic region known as IVD segments, we saw that the sharing of the Southern African Botswana was greatest with the Zambians. Also, East African Bantu speakers from Uganda were found to have highest genetic affinity with Zambia among all Central and Southern African populations. So several additional analyses, including admixture modeling, also showed Zambia as the best intermediate population between Central, East, and South African Bantu speakers. Moreover, in comparison to a route via DRC to Zambia, a route via Angola seemed more likely. Therefore, based on these results, we supported the view that Bantu migration or expansion to have moved south of rainforest present in Angola and proposed that it then moved eastward only after reaching Zambia, the eastern and the southern stream of Bantu speakers separated out. However, these findings need to be interpreted with caution as Bantu expansion was likely very complex, multi-wave phenomena, and much more fine-scale data is required to understand it comprehensively. And in this context, a recent preprint by Talman and colleagues indicate that Bantu expansion could in fact have been a multi-wave process instead of a single migration. So let's get back to the PCA. So in the PCA, the West African populations from our studies, such as one from Benin, projected in proximity, but did not overlap completely with other West African NC speakers, such as Yoruba and Isan from Nigeria, making some sort of continuum of populations from Nigeria to Mali. Interestingly, we observed BRN, the largest ethnic group from Plateau State region of Nigeria, to show a very interesting clustering pattern. The BRN samples localized independent of all other West African populations and slightly tending towards East African population. So we thought, could admixture be a source of this? So this is the admixture plot based on our data along with data from previous studies. Some of our populations are highlighted by black rectangles. So although it is difficult to see things in this plot, what is apparent is that despite our populations being predominantly Niger-Congo speakers, most of the other African ancestries are represented in our data set by admixture. So let us focus on what is this admixture plot showing for Peru. The admixture analysis, as you can see in these large cutouts, provide a rationale for the unique localization of this group in the PC plots. And we could see that among all the Central West African populations studied, the Perom was the only population to harbor a significant dark blue component. This dark blue component could be traced back to East Africa and specifically to Nilo-Saharans such as Kalenjin and UNS. This admixture ranged around 15% and was more or less consistent across the population. Although this is not the first time such an admixture has been reported in West Africa, this is the first report of such gene flow into central Nigeria, and that too in a very large population like Peru. As mentioned in the previous section, there are several methods of dating admixture, and we use the tool Molder to date admixture in the Peru. The East African admixture in this population dated to about 1800 years ago, which differs from previous dates for trans-Sahelian migration, and not only to the broader geographic, not only to the particular area, but to the broader geographic region, suggesting that this admixture could have originated from an independent demographic event, either at the local or even at a larger geographic scale. So now moving to variant discovery, among the 40 million variants identified in our study, about 8% were novel with respect to variant databases and previous sequencing efforts. To unpack how ancestry and geography are reflected in this novel SNV or SNP content, we compared the novel SNP counts normalized to 24 individuals per population group. Although Nigeria is the best studied African country in terms of whole genomes, the Bedouin from this country was found to have a large proportion of novel variants. And this emphasizes that the current Nigerian groups are not only poor proxies for Africa, but maybe poor proxies for Nigeria also. However, the novel SNV representation here was pale in comparison to what we saw in the Botswana. And Southern African admixture plots show Botswana to have significant Khoisan ancestry, which is shown in the screen component. And we also found a strong correlation between proportion of Khoisan ancestry and the number of novel variants detected in an individuals. We would also find examples of such correlations for other ancestries, but I'm not going there. So finally, we attempted to test whether novel variant discovery is reaching saturation in African populations. And in fact, if we conservatively start with the variant discovery in Botswana and ask how many more variants are discovered in each group, we don't see a plateau. And even if we limit this to common variants, we still have almost 6,000 variants between the last two populations, showing that we are definitely far from saturation. So moving to another aspect, so natural selection in response to pathogens have played a major role in shaping African genomes. And as you can see in this plot from a review by Fan et al., malaria has resulted in selection of variants in several genes such as G6PD, HPV. Similarly, there is a strong signature of selection on the APOL1 gene related to trypanosomiasis in some of the West African populations. And there are other interesting examples such as selection for height in the rainforest forager populations and for altitude in high altitude in certain Ethiopian populations. So to further assess the contribution of natural selection to the genomic landscape, we used a widely used estimate known as composite likelihood ratio, or CLR, in each of the 6 populations. The circular Manhattan plot, although it's very difficult to see, shows the selection signals in 10 kb windows across the genome. So the outermost is Botswana, then Zambia, Cameroon, BRN, FNB, and WGR. So, and based on all these, we identified about 107 in these populations, 62 of which were in novel genes. And a large proportion of these novel genes were involved in immunity, DNA repair, and metabolism. As we have seen before, immune genes are somewhat common in any selection scans, and several signals for immunity to protozoan diseases, such as the ones we discussed, such as APOL1 and HPV, are well known. However, what was surprising here was that a substantial proportion of the immune genes detected in our scan were related to viral immunity, suggesting that viruses might have also played a major role in shaping genomes, at least in some parts of Africa. And although almost half of the novel loci showed signs of selections in 2 or more of the 6 populations, several of the signals were population or region specific, as you can see in this plot. And even some of the well-known signals, such as FOXP2, showed high variability in selection strength across these groups. Given the recent shared history, these differences are intriguing and emphasize the necessity for more nuanced studies on selection across the continent. Finally, we surveyed the frequencies, some of the well-described disease-associated alleles commonly found in populations of African descent. So what you can see here is that the HPS, which is a signal related to malaria or allele related to malaria mortality, was found at typically high allele frequencies in malaria endemic West and East African populations. Interestingly, when we compared HPS in 2 populations from Uganda, the Bantu-speaking and the Nilo-Saharan, the Bantu-speaking shows the highest frequency in the continent, whereas this allele was almost absent in the US. And this was primarily due to the major ancestral divergence evident in the PC and the admixture plots, and this reiterates the critical role of fine-scale sampling of variants and population structure to characterize genetic diversity and disease susceptibility.
Thanks, Wilson, for playing this. So maybe, I think as we are already at the 40 minutes, so I'm wondering whether we should stop here and start the discussion. So Declan, how would you like to go about it?
Yeah, so thanks so much for that talk. Yeah, we can move on to some questions. If anyone has any questions they'd like to ask, you can either raise your hand on the Zoom or just pop the questions into the chat. Yeah, so I guess I can start off with a quick question. You mentioned about saturation on the number of, I guess, novel variants. Have you an estimate for, how many samples you would need to essentially sequence to reach that?
That's a very good question. So we haven't really calculated it so far. And what our, I mean, not only this particular study, but all our studies have shown is that it kind of depends on now. I mean, there are certain regions where we won't discover a lot, but then there are populations and regions which are really untapped. So, and if we, I mean, so it won't be linear, but it would depend on whether the samples you are sequencing have some deep ancestry, which is not included in the databases. So it might be like, I mean, but there is still a lot to discover. And I mean, here we are currently conducting the stage 2 of the study, which is now called Assessing Genomic Diversity in Africa, or Agenda study. And there we have handpicked certain geographic regions and population ancestries where there is almost no data. But so, I mean, with that study, once we get to those results, we might say whether, I mean, we can review this and say whether we are really closing on saturation or it's still a bit far. Yeah.
Okay, so a question has just come through. My question, in terms of admixture, I'm curious about Neanderthal, this admixture possibilities. You may have alluded to it.
Yes, so I think, I mean, there is a lot of debate on whether Neanderthal admixtures in Africa happened or not. There are certain studies. I think there was a paper by Skoglund and colleagues who really made a good case for this, but then there are other studies and preprints which are showing that it might not be true. So I won't really stick my neck out, but I would say keep watching the space. So we might know it conclusively in the next few years. So right now it's kind of unclear. And maybe also the other thing that I kind of wanted to talk about, but is that there might have also been some ghost populations which have contributed to different ancestries. So these are actually populations which are no longer exist as such, but their contributions to certain populations might still be seen. So this is another avenue where I think with more ancient genomes, we'll have much more clarity on how this internal population structure, like especially the early structure, was like. So yeah, so a lot is going on here.
So another question, great talk. Considering the impact of pathogens and diet on African genome diversity, what can you share on how far different genetic modification patterns have contributed to the evolution of modern human Africa?
Yeah, this is also a very interesting question. So thank you. Yeah, so I think we know that specifically for like a significant portion of the genome, it might have really driven it because there is no strong impetus as a pathogen, because especially a deadly pathogen means that you live or you don't. So there, I mean, they kind of drive it, but I mean, in terms of overall structure, it's difficult to assess. But if you are looking at certain genomic regions, definitely, I mean, we will find perhaps a lot more as we study more populations and ancient genomes.
Okay. Then kind of, so you found a lot of variants associated with the immune response. Are they under strong selection or?
Yes, so I think we found a lot of variants that are related to, that are in genes that are related to viral immunity and that are under strong selection. And we also found evidence for known ones such as like HPV and others. So yeah, so like, I mean, we also recapitulated the known ones such as the malaria ones and the trypanosomiasis ones. So, but I mean, we had planned to really dig deeper into this, but we haven't been so far. But I think there are discussions on more in-depth studies on selection on these and other data sets. So we hope that sometime soon we will have more insights into the dynamics of these selections that are kind of related to immunity, or whether they're causal or whether they happen to because of some other function. So we will have more clarity soon.
Okay. Is there a plan to make this data accessible for minor allele frequency analysis of the novel variants for those of us working on Mendelian disorders?
Yeah, so, hi Lisa, yeah. So good to kind of see you again. So this data is completely public. So you can just go to EGA and then you just need to apply via H3 DBAC. You can get it. But that's kind of still a more difficult or tortuous route. So we are developing two approaches. So one is called the African Genome Variation Database, where you can quickly query the frequencies of these SNPs. And also a beacon is being developed by the H3A Bionet, which will enable you to really look up your variant without going through any process. So I think there are different layers of access, but yeah, I mean, it's going to be, it's already easy, but it's going to be easier with time. Okay.
Okay, I guess if, I know we've one more question. Would you give us a link to the African Genome DB? Sorry, this is
yes. So I think the African Genome Variation Database is still under development. I'm not sure whether the prototype is public or not, but I think, yeah, I mean, as soon as it becomes public, it will be very well advertised, Hughes. So I think you will surely be able to know about it.
Yeah. And then one question from Chantal. Thank you very much for an interesting talk. I wonder about the lack of genetic data from diverse populations and the development and implementation of genetic tests. Until we have more information from diverse data sets that look at what we should consider as a way of correcting for genetic ancestry to determine disease associations, in particular thinking of polygenic scores, which currently have much popularity, but again, there is a lack of development in diverse data sets.
Yes, so I think this is a long question, and I mean, I'm kind of trying to understand the sense. So I think this is Chantal. So Chantal, if there is, if I go off, just let me know. Then I will come back to the track. So I think, I mean, in general, we need to use PCs anyway, because I mean, even if you, I mean, for all GWASs, especially with multiple African populations, you need principal components. I think that's also true for polygenic risk scores. But then, as you have mentioned, other approaches, especially such as admixture, I mean, we need to be aware of that and try to somehow control or adjust for that to make our predictions precise. Otherwise, I mean, simply running this on the data without awareness of the deep structure and the nuances, especially certain ancestral components, which might not be really relevant in the admixture plot, but they might still be driving up the differences in some of the alleles. So I think it's very important that we need to do a very good characterizations of the population before we embark on disease variation studies. Hope I got it.
Thanks for tackling that. Okay, if there's no more questions, then I guess we can end the webinar here. So thank you once again. That was a fantastic talk.
Thanks so much, and nice meeting you and everyone in the audience. So thanks.
Bye.